Thank you everyone so much for joining us tonight. We'll be getting started momentarily. Thanks for joining us this evening. We'll be getting started in just 1 minute. Hello and good evening to each of you. Thank you so much for joining us tonight for Ask Me, our educational webinar series. My name's Audra Hines, Vice President of Care Services for the Golden West Chapter of the ALS Association. Today, June 21st, is Global ALS and Motor Neuron Disease Day. All of us at the chapter are so pleased to be here with you as a part of the International Alliance of ALS and Motor Neuron Disease Organizations to recognize and support the rights of people living with these diseases and to raise awareness about their impact. We could not make programs like this one tonight possible without generous donations to the Golden West Chapter, and I want to thank our lead sponsor for the Ask Me series, Mitsubishi Tanabe Pharma America, as well as our special donors, including the Ron Malone family and the Morris family. As a part of best serving the country's largest population of individuals living with ALS, at the core of the Golden West Chapter's mission are 3 interdependent priorities. The first is ALS care services, defined by the multi-layered approach that our care managers provide, including support groups, multidisciplinary clinic support, individualized one-on-one engagement, emotional support, and equipment loans. The second priority is ALS advocacy, which is focused both on individual, client, and family advocacy, all the way up to our involvement in federal and state legislative activism. The third priority that we're focused on as a topic for tonight is ALS research. In addition to the close relationship that the Golden West Chapter has with the California Institute of Regenerative Medicine and our advocacy work around helping them secure funding over the last few years, members of the Golden West Chapter leadership team have been appointed to several volunteer roles within the Institute, including serving as a part of a clinical trial advisory panel and on a committee that provides recommendations regarding funding and ethical standards. And we hosted our 12th annual California ALS Research Summit this past January, which was the highest attended summit the chapter has ever seen, with some amazing thinkers and innovators in the field. The summit is planned by researchers for researchers and brings together investigators, clinicians, biotech companies, government representatives, and advocates in ALS. And we also launched our industry partner consortium that supports funding for ALS research efforts, thanks to our very strong partners and supporters. To learn more about ALS and the Golden West Chapter, please visit alsagoldenwest.org for more information. As a part of our commitment to you and wanting to understand more about what really happens inside a research lab, we tapped on the shoulders of one of the top experts in this field. Here with us tonight is Dr. Justin Ichida, who's the John Douglas French Alzheimer's Foundation Associate Professor of Stem Cell Biology and Regenerative Medicine at the University of Southern California. He's also a New York Stem Cell Foundation Robertson investigator and has been a strong partner and collaborator with the Golden West Chapter for many years. Dr. Ichida is a member of the California ALS Research Network and served as a local host for our annual research summit when it was held at USC. He's given many educational presentations throughout our service area in both California and Hawaii, and has been a longtime participant and supporter of our Walk to Defeat ALS. Dr. Ichida was honored with our ESSE Commitment to a Cure Award for his outstanding achievements in the fight against ALS. Dr. Ichida, thank you so much for being here tonight. We're thrilled to have you.
Oh, I'm thrilled to be here. Thanks for having me.
Absolutely. Thanks for taking some time. And I want to let the audience know before we move further into the presentation, we wanted to actually show our audience a real ALS research lab. So thanks to our strong partnership with the USC ALS Clinic and with its researchers, Dr. Ichida agreed to give us a little tour and a little glimpse into the inner workings at his lab. So let's take a look. Thank you so much for being here with us tonight. We are here at USC with Dr. Justin Ichida at his wonderful lab doing all kinds of ALS research, and we are just outside the tissue culture room. We would love to hear more about what happens in this room.
Certainly. One of the key challenges in understanding how ALS kills nerve cells within patients is that the types of ALS are very diverse. There are many different causes, and we need to understand all of those causes while also finding therapeutics that will work against many different types of cases. So we've developed a technology that allows us to take blood cells from ALS patients and convert them into stem cells and then nerve cells, which then mimic the disease in a dish. And so that's what happens largely in this room. Once we have those disease models, we call it, for different ALS patients, we then zoom in to find the actual molecular changes that are occurring, why the nerve cells are dying. And then we can also test both approved drugs or novel compounds or drug candidates to try to stop the nerve cells from dying. And that all happens in here. This room is the Choi Family Therapeutic Screening Center here at the Broad CERM Center. What we do here is high throughput drug screening. So this is one of the rooms where we can test all approved drugs or any new drug candidates for the ability to slow the ALS nerve cell degeneration. And so once a day, this robot arm will take the cells out of this incubator and move them into this imager, where this imager will image every single millimeter of this plate. And so we get an image of all of the cells in the dish. This is a time lapse of about 30 days of imaging. You can see here we have motor neurons on this side. So these are the spinal cord motor nerve cells that are the ones that are vulnerable to ALS. On the right side, we actually have muscle cells. And what we're looking for here is just how long these nerve cells survive and how well they project their axons to the muscle to connect. So this is the connection that basically degenerates in ALS patients. And so we're mimicking that here and looking for drugs that promote that connection. And so that's the main setup here and the function of this room that we have that's quite unique here at USC.
Dr. Ichida, a couple other questions we had. I wondered too, how do you, when you're picking what to research specifically in ALS, how do you collaborate with other ALS researchers so you know there isn't too much duplication of efforts?
Yeah, that's a great question, Audra. I think today's science is largely driven by collaboration. Mainly, I think it's because, one, people have recognized that if you work together, things move faster. Two, there's more funding available, especially for neurodegenerative disease research from folks like the ALS Association and others, that allows you to not have to compete for funding, but but go for awards that require collaboration. So that's important. So here we're using a confocal microscope, which allows us to magnify an image by over 100 times. This allows us to see individual nerve cells at a very high resolution. So you can see here, this is a motor neuron from an ALS patient that we've made in the dish. And what we're looking at is the nucleus, which contains all of the DNA. These are the neurites that allow the motor neuron to connect to other neurons and to muscle cells. And one of the important things to look for is the protein aggregates that can sometimes form out here in the cytoplasm, which you can see in green. And so these are things that these little green clumps are what we want to get rid of in the ALS patient nerve cells to keep them alive. So this is a way we can actually study that process in real nerve cells.
And we'd love to hear specifically about your work right now. What are some things that you guys are investigating?
Right, we have several things that we're very excited about. I'd say one of the most exciting things is that we found a new way to induce nerve cells to get rid of the protein aggregates that are driving their death in ALS. And so this is a pathway that people have not tapped into before for ALS treatments. And so we're excited because it seems to, in the dish at least, outperform all of the existing drugs for ALS. The second thing that we're really interested in is the fact that the immune system of the brain seems to be playing a huge role in affecting the progression of disease. And what we've understood over the last few years is that there are the immune cells of the brain are called microglia. They exist in different states, and in many ALS patients, they exist in a state that actually drives the death of neurons instead of protects them. So our new strategies are to actually reprogram the state of these microglia to become protective toward neurons instead of toxic. And so that's something I think has a large ability to extend the disease survival time and really slow down the disease once it's already started.
I just want to thank you so much. This has been so educational and informative for me. I'm sure it is for our audience too. You're doing amazing work here, and it's going to be tremendous for people with ALS and people with ALS in our community. So thank you for opening your lab and letting us poke around a little bit today. This has been amazing.
Yeah, thank you for coming, and thank you for all the support that the Golden West chapter has given to us over the years. It's just been an amazing partnership. Establishing the Certified Center of Excellence has really helped us to, jumpstart the research, and anything we can do to help patients understand, what we're doing and how hard everybody's working on their behalf, I think is great. And thank you for, coming to showcase that.
Thanks, Dr. Ichida.
Thank you.
We're back. That was great. Thank you so much, Dr. Ichida, again, for letting us spend a day with you. It was just very cool to meet your team and walk around the facility and know that real change is happening there. It's awesome.
Yeah, that was fun. And good job on the video. I think it looks cooler in the video than it does in real life.
Thanks to the editors for that. So I think our audience would love to hear a little bit more about your work if you have a few slides to share.
Sure, yeah, I'd be happy to do that. Let me see if I can share my screen, and I'll try to go full screen. So yeah, what I thought I'd do is just share, a few slides about what we do, just to give you a better understanding of some of the modern approaches that we and others are applying to try to find new therapies for ALS. And we're really coming at this through the lens of stem cell technology, which I think has been a game changer for the entire field. And I'll try to explain why. First of all, our primary motivation is, of course, you, the patients. We were very fortunate to be able to get to know Nancy Ryder over the past several years. And she really inspired us because she spent, so much of her time trying to do things to promote awareness, ALS awareness, and also further research and care. Even though she knew that she wasn't likely to benefit from these things because of how long it takes to actually develop new therapies, but she gave so much of her time, even when she didn't have that much of it left, to be able to help future ALS patients. And that just really inspired us to do as much as we could and work as fast as we can to try to find some new therapies. And so I'll tell you a little bit about how we're doing that here. As you know, there aren't enough effective therapies for ALS. And one of the critical reasons we believe this is the case is because in reality, ALS has many different genetic causes. And so the issue is that, given that ALS is a little, is relatively rare, to get enough patients together for any given clinical trial, we usually have to include many different types of ALS patients. And I think some of the data is coming out now where they're indicating that indeed it's actually, in most cases, I think what a given drug is not that likely to work for all forms of ALS. And so we really need to find, new therapeutic approaches that are able to work for more types of ALS patients or all ALS patients. And that's kind of what we developed a stem cell platform to do. And so to do that, we wanted to create or use, ALS patient nerve cells in the Petri dish to be able to screen many different types of drugs and drug candidates to find the rare, compounds or drugs that could improve the survival of these nerve cells, regardless of which ALS patient they came from. And so, however, to biopsy nerve tissue from a living person is very difficult. And so we decided to use a stem cell technology to get around this problem. So the way the stem cell technology came about is when we were able to take blood from many different ALS patients. And so first of all, I'd really like to thank all of the patients in the Golden West chapter group that really donated their blood for this. We took those blood cells and we essentially changed the identity of those cells into stem cells, which we could then expand and have store banks of in our freezers. But also we could make those into any type of cells in the body. And we chose to make them into nerve cells, the nerve cells that are lost in ALS. And what's kind of, amazing about that is that once we make those nerve cells, because all of the DNA of the patient is preserved in those nerve cells, they usually begin to manifest the signs of ALS, or essentially what the nerve cells in an ALS patient would look like. And so one of the things you can see is that they start degenerating faster than those from people that are not affected with ALS. And we use this kind of change to screen or find, test thousands of different drugs to find ones that could improve that survival time of those nerve cells for not just for one patient, but for all of the patients. And so that's what we did in the past few years. And this is just some data, kind of just showing what that survival time looks like in real neurons. So these are nerve cells. All these black lines are following nerve cells from different people that are not affected with ALS, and then these red lines are following the neurons and their survival in the dish from 3 different ALS patients, all with the same form of ALS called C9orf72. And then on the right is a separate experiment where we did the same thing for, again, 3 unaffected people here, but then also 6 different people with sporadic ALS. And you can see in both the C9 ALS and sporadic ALS, we see this very rapid decline of survival of these nerve cells made from these people in the dish. And this is, similar to what you would see in the body, but a little bit more accelerated. And so that's the one difference we used. And to really convince ourselves that we were actually looking at ALS in the dish, we looked even at a more microscopic level at the protein aggregation that many of you are probably aware that kind of occurs in the nerve cells of ALS patients and is actually driving the degeneration of these nerve cells. And that protein that's of interest here is called TDP43, and it's usually localized in the very center of the nerve cell here. But in ALS, it leaves that central area and moves out to the side of the cell here, and it also forms like a very discrete aggregate. And that process is what causes the nerve cells to die. And there are many different reasons for why this can happen. But we can see the same thing if we make nerve cells or induced motor neurons, we call them, from patients, and we put them in the petri dish. You can see that in an unaffected person's nerve cells in the petri dish, that TDP43 protein is all localized in the center of the cell. But in the patients with C9 ALS or sporadic ALS, when we make their nerve cells in the dish, you start seeing the TDP43 moving to the outside of the cell, and essentially that is the disease process that is occurring in real life in patients. So we felt pretty confident that this was a real representation of what's happening in ALS patients, and so we thought that this would be the first opportunity to really test all approved drugs, as well as a couple thousand drug candidates, to see if any of them could improve the survival of nerve cells made from all of these different ALS patients. And Yichen and Michelle and Gabriel in my lab kind of teamed up on this project. I want to point out that this project was actually funded by the Department of Defense. And the Department of Defense decided to allocate money for ALS research every year based on the lobbying of several important agencies, one of which, of course, is the ALS Association. And the ALS Association was really instrumental in getting the Department of Defense to do this, but they completely funded this collaboration between USC and Sanofi, the therapeutics company, to do this big screen experiment. And essentially, we had different types of neurons from different types of ALS patients. We treated all of them with all of these different drugs, so thousands of drugs. And then we found the ones that made the nerve cells survive longer for all of the different types of ALS neurons. And that can be distilled in this kind of chart here. And essentially, these are only the drugs or drug candidates that worked on multiple lines, on neurons from multiple ALS patients. And just to kind of orient you, each vertical column is actually neurons from a different ALS patient. And on the horizontal rows, those are different drugs or drug candidates. And if there's a red square, a red rectangle, that means that there was a positive influence of that drug on these ALS patient nerve cells. So what's interesting is that all 3 of the patients here on the right are actually the same type of ALS. They're the C9orf72 type, and you can see that their nerve cells all responded very similarly to these drugs. But what was very striking was that if you look at the rest of the ALS patients, you can see that most of their nerve cells did not respond positively to those same drugs. And so that is really a representation of, I think, one of the challenges of ALS clinical trials, which is that there's so many different types of ALS patients in the trial. Most of the drugs don't have strong efficacy across all of those different patient types. But some good news is that we did find a number of drugs or drug candidates that were effective across almost all ALS patient nerve cells. And those are the ones that we've been exploring further. And I'm going to tell you about one of them here. So this is one drug candidate that had one of the strongest effects we've seen on these patient nerve cells. And this is just an image of what you can see in the Petri dish. All of these white spots and with these extensions are different nerve cells from an ALS patient. And you can see with the placebo, they die off pretty quickly. But with this drug candidate, we have a large amount of them left at the end of this study. And so this is a candidate that works well for all types of ALS neurons. And we wanted to see if we could test this, move this into the clinic. But the problem was that it's a cancer drug. And while it's very active on ALS patient nerve cells, it doesn't get into the brain. And so we needed a way to make basically a brain penetrant version of this drug to see if we could move this into clinical trials. And so with the help of some ALS Association funding, we actually were able to make a new version of this drug that now reaches the brain. And this is an example of what the effect looks like in a mouse version of ALS. So these mice both have a genetic mutation that causes them to have ALS, and they normally have very difficult, very limited mobility. But in one case, one animal we injected with a placebo, and the other one we administered the drug that we made with this in the ALS Association project. And you'll be able to see the difference in mobility. It's marked. And this longer tailed animal, this mouse got the new drug, and this one on the right with the short tail didn't. And so this is, hopefully this is representative of what happens when we go into clinical trials. But I think one important thing to say is that because of this project and moving this along, we've now been able to essentially license this drug candidate to a biotechnology company who is now testing it in larger animals for safety, which is the last step before taking it into clinical trials from humans. So really, it's been a process of going from a very early research project in the lab funded by the Department of Defense to a more mature project funded by the ALS Association, and now it's left the university completely and is in the biotech sector, hopefully going into patients in 2023. So I think that's the last slide. And I don't think it's critical to know, but just for your information, I talked about this, pathology, this protein aggregation that happens in ALS patients. And in these mice, we see essentially complete reversal of that pathology. You can see in the ALS mice, they lose that protein from the center of the cell. But when we treat them with the drug, it restores it. And so both the survival of the neurons and the protein aggregation is sort of mitigated. And also this translates into the animal moving around as a normal animal would. So I think that's the end of the presentation. Let me know if you have any questions.
Thank you so much for that. That was great, really helpful. We do have a few questions. Some people who are here tonight submitted a question ahead of time, Dr. Ichida, so I'm going to ask you a couple of those. Okay. One person is wondering, asking, what are some of the high potential clinical trials going on right now? They say they live in the San Francisco Bay Area. Do you have a sense of what to tell them about that?
Yes, I think, well, certainly the Amylyx combination is something that, looks very promising. And I think it's probably, in our hands. I didn't show that data here, but it is also, that combination is one of the things that is effective on most patients' nerve cells in the dish. It's not maybe as effective as some of the other things we found, but it is very broadly effective. And the data in the clinic look reasonably promising. I think with a longer trial or more patients, it would be even more significant. But it looks like in Canada they approved it, and the FDA in the United States is taking a closer look. So that's something that I think is out there and reasonable to do. And then the others, some of them depend on the particular genetic mutation that somebody might have, if they do or do not. But there are a couple of trials. For example, there's one called the ataxin-2 antisense oligo trial that's occurring. That's a Biogen trial. That might be one to think about trying to enroll in as well. Yeah.
Great, thank you. Thank you so much. Another person's asking about any new research potentially on gut health and ALS.
Yeah.
Specifically, if there are any studies that have shown that ALS patients process food or nutrients more poorly than relatively healthy people.
Yeah, I don't, that's a good question. I don't know about processing nutrients more poorly, but what's kind of fascinating is that this hasn't been, well, yeah, so there have been studies in the past few years showing that in other types of neurodegeneration, for example, Parkinson's disease, the exact composition of the gut bacteria can have a big influence on Parkinson's disease progression, at least in the laboratory. And the same has been shown recently for at least one form of ALS called the C9orf72 form. The gut kind of composition of bacteria can greatly affected the health of mice that kind of mimic the C9orf72 version of ALS. These mice, I'll say, like, these mice were more severely compromised based on their how they genetically engineered them to mimic ALS. I think they're a little more severe than the actual patients. So it's not 100% clear that would be the case for actual ALS patients, but I think that there is some good early research out there saying that the bacterial composition in your gut can actually have some important effects on overall health, and so especially in the context of neurodegenerative disease. And so I think it's, I don't know if we have like a magic bullet on how to actually leverage that information yet, but it's something to, I think, read as much about as we can, and then see if we can make some sense out of it now. Yeah.
That's terrific. I think this person's asking this question in relation to your presentation, asking, will this treatment, when available, be available effective for PLS patients? Would it prevent possible progression to ALS?
Yeah, it's interesting because it's currently very hard to tell the difference between PLS and ALS, at least in early stages. And the same same protein aggregation that I showed in the slides, that exact thing also occurs in PLS. And so because the way that the drug works is to clear out that type of aggregation, I think there's a high likelihood that it would also work for PLS patients, and if it works for ALS patients. So yeah, because of the conserved mechanism of disease, I think there's a high likelihood that it should work. Yeah.
Okay. And related also to the work you're doing, Dr. Ichida, how do you give an animal ALS? And if you can give it, doesn't that show you how to take it away?
Yeah, it's a good point. We basically, for that animal, you give it just a slightly higher level of that TDP43 protein by just adding an extra copy of that gene to the DNA of the animal, and that triggers all of this aggregation that occurs. And so what's really tricky about that protein is that we do have good methods for for example, reducing levels of the protein, even in humans now. But it's what I didn't really explain is that that protein causes toxicity to nerve cells, both by aggregating, and also when it aggregates, it loses its normal function. So losing its normal function is also a problem. So you can't simply just lower the levels of the protein. You actually have to keep the levels kind of normal, but keep it from aggregating, and that's a more difficult problem. In some cases, ALS patients have genetic mutations that we know of, and so I'm sure by now probably heard of the CRISPR technology that allows gene editing. There are a lot of folks trying to use that gene editing to basically normalize or restore the proper genetic code to patients with an ALS causing mutation, and that's just going through the technical hurdles of delivering that kind of gene editing machinery safely to all of the nerve cells that need it. And that, there are a lot of people working on that. So presumably, in the future that will be possible. Yeah.
Terrific. We have someone else who's asking, is there any sort of correlation between the type of care the ALS patient receives and their responsiveness to the medication they're taking? Such as the example they're giving, if an ALS patient who's frequently hospitalized or at a skilled nursing facility, comparing that to a person who's being taken care of in the home,
Yeah, I think that, one of the things that the ALS Centers of Excellence ALS Association Center for Excellence have shown is that the clinical care actually makes a substantial improvement. It gives you a substantial improvement in motor function and lifespan once the disease occurs. And that's like real, human data. So that is really important. And I'll say that we see something very similar in these animal studies. So in these animal studies, these animals, as they become less and less able to move around and closer to paralysis, they actually have a hard time reaching the food and the water. And so they get severely malnourished and dehydrated. So we do a lot of things to keep them hydrated. We put a lot of gels in the cage and a lot of also sort of like jello with nutrients for just regular nutrition. And that actually improves their survival quite a bit, but also allows the drugs to work a lot better. I think when we don't do that, they start dying from other things that aren't really ALS per se, but malnutrition or dehydration, and that and then in those circumstances the drugs don't work so well. So I think patient care is a really critical component. That's why I think having both research and clinical care being heavily invested into is a really good decision for example, the ALS Association. Yeah.
Terrific. Thank you so much. Is ALS a pure motor disease, or is the brain involved? Is there any research in this area?
Yeah, there's a lot. That's a great question. There's a lot of research in that area. And because you kind of always think of it as a pure motor, or people say it's like a pure motor disease, but it's increasingly clear that while many patients may have only motor symptoms during the time that they have the disease, there are other patients that have combinations of motor difficulty and also cognitive changes. And we now know that certain genetic mutations or certain genetic causes of ALS are ones that have a high likelihood of also causing dementia. The C9orf72 version of ALS is one of those examples. So it's not purely a motor. It's, I guess I would say there are probably 80 to 90, maybe 80% of patients for which it is a pure motor disease during the whole time that they have it. But it's really not uncommon to find ALS patients who also have a cognitive, are affected cognitively, unfortunately. Yeah.
Okay. And related to that, similar. Someone's asking, how does someone know what their form of ALS is?
Yeah, that's a good question. I mean, I wish we could better do that. I think there are some interesting approaches coming up. So the most, probably maybe the most definitive method is the genetic sequencing. And there are groups out there trying to make this, which is their mission is to make everybody, make it so that any ALS patient can have their genome sequenced for free. And, you know, for example, Synapticure is one of them trying to do that. But there are others. And I think that's one way to do it. So for about 10 to 15% of patients, you can figure out essentially what type of ALS you have based on that. The other 85%, it's quite difficult to know. You know, I think what we're moving towards is I guess the reason is because for most of those other patients, either they have a very, very rare form of ALS that we haven't really verified as a form of a cause of ALS, or they may have a combination of different genetic changes that combine to cause ALS, and that's why we haven't been able to kind of like pinpoint all of those genetic combinations as a particular form of ALS. But you know, unfortunately, at this time, only 10 to 15% of patients will know what form they have by the genetic sequencing, and beyond that, it's actually quite difficult to know. So I think like one of the things that we're trying to kind of move towards clinical treatment is this type of system that I kind of presented, which is like the nerve cells in a dish. If we can figure out how to make this available to all patients, and make it, you know, faster, then we could theoretically at least have some idea of what drugs they might respond to before they enroll in certain clinical trials, and that I think could really increase our chances of success in most cases. So yeah, that's kind of where. So there are people like really driving on the genetic side,
Yeah.
And there are people like us moving on the cell modeling side. Yeah.
Right. We need both. Yeah, makes sense. I do have one more question. I just want to remind everyone, if you have a question for Dr. Ichida, just please go ahead and type it into the Q&A function for him, and I'm happy to ask it out loud. Someone is wondering about TDP-43, if there are any epigenetic factors that drive the protein to migrate out of the neuron's membrane. Technical question.
Yeah, that is really technical. I think that is very likely. I think that you know, the reason why the protein moves from the center of the cell to the outside is not very well understood. I think that there are one thing is that the little inner cell border that keeps things in the center of the cell can deteriorate, and that's why the TDP-43 leaks out to the side of the cell. But other things could be that it in the center of the cell there are specific you know, RNA and protein products that it interacts with, and the epigenetic changes that this person is asking about, you know, these epigenetic changes basically change the kind of complement of RNAs and proteins that are in that center of the cell. And so if you change it in a way that the TDP-43 protein is now no longer able to interact with those RNAs because they're not there, that could cause the TDP-43 to leak out into the sides of the cell and cause damage. And so that could very well be the case. And that's really why I think ALS has been so challenging to treat, is because there are all these different ways of causing the ultimate problem, which is that the TDP-43 kind of moves out of the center of the cell into the outside. And so you could find a treatment that works well for one of those ways, but not for all of them. And so I think what we're doing now is finding ways to kind of address that central problem, or the most sort of downstream problem, which is the TDP-43 moving out of the center of the cell. and that should translate into more effectiveness across more ALS patients. So, that's what we're hoping.
Gotcha. Is there such a thing as stem cell therapy for ALS?
Yeah, for sure. There are a couple of things, there's kind of a neuron trial of some, transplantation of cells. At Cedars-Sinai, they have transplantation of kind of neuronal neuron progenitor cells that secrete protective factors for nerve cells. There's a group from Israel who is basically started with stem cells, and then they made the supportive cells of the brain called astrocytes. And I probably shouldn't even call them supportive cells, but they are classically known as supportive cells, but they actually do a lot to help the function of neurons. And so they make these stem cell derived astrocytes, and they're transplanting those into ALS patients, which I actually think makes a lot of sense scientifically. A lot of the problems that you see, at least when we study animal versions of ALS, is that the astrocytes and non-neuronal cells become completely aberrant or unlike how they should be. And we have documented cases where in animals where those astrocytes and non-nerve cells are actually causing the death of the nerve cells. And so I think that's a strategy that could work. And so we'll be following that closely to see how that happens. And we do have our own version of that type of program. We're not making astrocytes, but we're making other cells called microglia, which are the immune cells of the brain. And we want to transplant those into patients to see if that will, mitigate disease.
Thanks, Dr. Ichida. I do have one more question. This is coming internally from someone at our chapter. The Golden West chapter is considering a plan to have each person diagnosed with ALS be given the opportunity to have a full genome scan and to donate blood in support of ongoing new research. Yeah. So the question to you is, how would having the genomes of a couple thousand people, plus about 40 new people that are registered with the chapter each month, living in California and Hawaii, how would that accelerate research?
Oh, this is a great question. Actually, if we could, or if you could execute that study, it would have, I think, really a big impact. And the reason is because one of the things that's unique, there are many things that are unique about the ALS Association Golden West chapter, but you have a large, group of patients, and you follow them over time, which takes a lot of work, but it's very valuable information. A lot of the genetic studies, assuming that we sequenced all the patients' DNA, and we have their blood for doing the types of petri dish studies that I talked about today, if you follow their progression over time, then we can ask the most critical question, I believe, which is, what changes in the DNA of people make the ALS patients live longer once they have ALS. So all of the genetic studies now, or the vast majority of them, are focused on what is likely to make a person have ALS, or maybe not have it in the first place. But that's not exactly the same thing as what DNA change makes them progress slower or faster. And the reason is because people just don't have that information. They don't have the information on many patients and how they progressed over time. But if the Golden West chapter could pull that off, then we could find potentially, new DNA changes and therefore proteins that we could target with a new drug that cause ALS patients to live for a much longer time with the disease. And so to slow it down to the point where it hopefully doesn't really progress. So that would be that would be the major outcome that I could imagine. So it would be a big effort, but you have a large, group of patients, and you keep track of everybody very carefully, and that's really the key.
That's great. We're certainly invested in finding ways to help you all as the experts in ALS research. So a good conversation to continue.
Yeah.
I don't see any more questions in the Q&A function, but if anybody has a last one, I'm happy to wait just a moment. Dr. Ichida, I want to thank you very much for all of your time and taking us under your wing in your lab and spending a little bit of a day with us. That was really fun.
Oh, thanks for making that video. Yeah, I gotta watch that again. Thanks for
Watch it when you have more time. You're not under pressure on camera. We do have a couple last quick questions. Well, this is more a comment. Slow progressors are excluded from studies, and it seems they could be very helpful going forward using their data within research. Anything to say about that?
Yeah, that's really, it's really unfortunate. And of course, like, we understand why, therapeutics companies do that. It just it's actually pretty complicated in the sense that I think the FDA believes that it's not really ethical to keep patients on placebo for very long. So you really need to have a relatively short clinical trial. And therefore, it's hard to actually see a difference in, an effectiveness of a drug because the time is relatively short. And so in reaction to that, people, therapeutics companies will enrich their clinical trial cohort for the fastest progressing patients, so they have the greatest chance of success, essentially. Bigger signal window. So that's it's a tricky situation there, and it's really, it is unfortunate that's a downside of the people that progress slowly. But you're very right that I think there are some important pieces of information in the DNA of those people, and that's what so I think that study that person, the previous questioner had proposed would be a great thing to do. It just have a tremendous impact, I think.
And another question: why do you think the C9 ASO failed?
That's a good question. I think there are two possibilities. One is that kind of a simple possibility, which is that the drug that Biogen and Ionis made at a high concentration it just could be toxic, not because of because of like off-target, they call it off-target effects, but basically side effects that occur that are not because it's hitting the intended target, but sometimes they hit unintended targets. And that could have had some toxicity in the patients. And because it was only the patients at a high dose, well, none of the patients had an improvement, but the patients at the very highest dose actually trended towards declining a bit faster. So there could be some unintended side effects that the particular drug candidate had. The other possibility is that what we understand about the mechanism wasn't really correct. So the research leading up to it wasn't telling us the full story. So for a drug to have made it to the phase 1-2 clinical trial now, that means that when the gene was discovered in 2011, that year people started making this drug because it takes like 10 years for that transition to occur. So people made that drug without knowing a lot about the disease mechanisms, and then now we see the readout of it. That's just the unfortunate kind of experience that we all go through when making therapeutics. But it's important to it's important because you want to make sure that whatever you're putting into people is safe and hopefully not going to harm them.
Right.
So that's what takes so long. But you know, I think the second possibility is essentially that the mechanism that we're I don't think that the mechanism that they tried to achieve was wrong, but there may be additional things that they didn't really understand that counteract the benefit of the good thing that they did.
Yeah.
That's really the most likely explanation in my mind. So
Okay.
it's nobody knows for sure yet.
That's right. And final question. I'd love to just ask you, for our audience's benefit, what made you decide to go into ALS as a focus in your research?
Yeah, it was, you know, it's really kind of funny how things work out, but we were really just interested in kind of like synthetic, you know, biology. That whole kind of story that I presented about how we take blood cells and turn them into stem cells and nerve cells. At first, we were just really interested in doing that because scientifically, that's really cool, you know, that you can do that. You know, like 20 years ago, nobody knew that you could take a blood cell and turn it into a motor neuron or something like that. So we were just really interested in that. But once we kind of answered all of the interesting scientific questions about that, you know, we were really just left with this ability to make nerve cells from humans. We thought, what can we do with this? And, you know, then we started really realizing that the ALS, you know, ALS is just a huge unmet need, and we have the technology that I think could transform the way that we do drug development in ALS. And so we just went 100% into that. And it's been probably the most rewarding experience that many of us have had, I think. And hopefully when we actually, you know, make an effective therapeutic, that'll be, hopefully that's soon. But, you know, that's kind of how everything happens.
Yeah.
Yeah.
So interesting. Dr. Ichida, we're so proud and happy to collaborate with you. We feel so lucky that you're part of our chapter service area. Thank you so much for being here tonight. It was really helpful. I appreciate it.
Thank you.
And I really want to thank everyone on this call for joining us tonight. We're grateful to each of you for your time and commitment and just for being a part of our community. And once again, a big thanks to Mitsubishi Tanabe Pharma America for their support. I want to remind you, we have lots of other good stuff coming up in August and September. Mark your calendars. This includes our next segment in our Ask Me series, which will be on estate planning. And for the golfers out there, we have the Peterson Golf Classic coming up on September 12th, as well as the kickoff to our walk season with events in Hawaii and the California Central Coast, and our Napa Valley Ride, Hike, and Walk. So please do join us for any of these fun-filled, family-friendly events, either in person or via our virtual options. We've got something for everyone. And as you know, philanthropy fuels everything we do, and we're grateful to committed donors who give generously throughout the year. To learn more about how you can support these valuable programs we offer, please visit our website at give.alsagoldenwest.org. And please watch for the short survey on tonight's presentation that you'll receive via email. Thank you so much again, and we'll see you next time.